Well, good evening, ladies and gentlemen, and thank you very much for the opportunity to present to you at your virtual beef conference tonight. For those of you who may not have met before, my name is Andrew Bryne, and I'm the current Breed Development and Extension Manager at Angus Australia, which in a nutshell means that my role is to oversee the implementation of the different genetic improvement technologies which are available to Angus producers in Australia. My session tonight is aptly called Why Single Step is the Bomb, a topic which was allocated to me by Max Tweedy. But there are several things which I'd like to share with you. To go back firstly and during this session discuss the role that genomics or DNA technology plays now in making genetic improvement within a beef breeding program. To spend most of the time talking about the new technology, the new single-step software, which was implemented in December or late 2017, but into the December 2017 Angus BREEDPLAN evaluation for analysing genomic information. And lastly, to go back and discuss, well, what does this actually mean for the utilisation of genomics in a modern kind of beef breeding enterprise? Before we get into talking about genomics as the latest technology, I just want to spend some time, I guess, reviewing some of the first principles of breeding. And in particular, if we're looking at making genetic improvement within a beef breeding program, there are a couple of key ingredients for success. The first one of those is that we need to be able to accurately describe genetic differences between animals. And then secondly, we need to be able to use that description of those genetic differences to identify those animals that are most aligned or have genetics which are most aligned with our breeding objective, and then to use those animals to breed the next generation. Now that can be easier said than done, and in particular it can be quite challenging to identify and describe the genetic differences between animals. But thankfully in the modern age we have a range of different technologies and tools which are available to us which help us to describe genetic differences between animals. And so like in all aspects of our farming today, we have different technology to help us. In the case of what I have on the screen, in the case of the Angus breed, we have Angus BREEDPLAN EBVS. So the Angus Australia and New Zealand Angus Association conduct a joint genetic evaluation utilising software provided by ABRI and developed by the Animal Genetics and Breeding Unit, both organisations in Armidale, so software known as BREEDPLAN technology, where every fortnight we go through and produce breeding values on Angus animals for up to 25 different traits. And there are some subtle differences obviously between the breeding values which are produced on Angus animals in Australia and New Zealand, but the general concept is the same, that we're utilising technology to go through and describe the breeding or differences in genetics between animals, their breeding value, which we can then use to identify those animals which are most aligned with our breeding objective to use within our breeding programs. Now if we look at what goes into calculating those breeding values, there are several main sources of information. There's some very sophisticated technology which we're very lucky to have available to us. But then there's sources of information around each individual animal, such as its pedigree information, its performance information, and now genomic information, which goes into giving us a good estimate of those animals' breeding values or genetic differences between animals. And tonight I'm going to spend most of my time, or all of my time, talking about the third component of that, being the genomic or the DNA source of information. So genomic information is included into Angus BREEDPLAN, and it's not something new. We have had genomic information included into the Trans-Tasman Angus genetic evaluation since April 2011. And there are several different genomic products which we incorporate information from, being the two products provided by Zoetis, being the HT50K and I50K products. And more recently in Australia, the availability of the Angus GS product, which is provided to us by Neogen. Now there's been quite a large uptake of genomic technology within the Angus breed. If I focus here on the animals that are recorded with Angus Australia, which have genomic information recorded and by birth year, we can see now with the red bars, you can see the number of animals which have genomic information born in each year. And so in the current year, in 2017, we now have just over 10,000 animals which have genomic information recorded. Cumulatively, that means we now basically have around or just under 50,000 animals which have genomic information available on them to utilise within the Angus BREEDPLAN genetic evaluation. Now this slide here only describes the animals recorded with Angus Australia. There is of course several thousand animals, as I understand it, with New Zealand Angus, which also have genomic information, and that is growing as well. So it's quite a large amount of genomic information now, which is adding significant accuracy to our EBVs. I guess along those same lines, why do we incorporate genomic information in the calculation of EBVs, or our breeding values? And the reason is really around trying to produce breeding values which more accurately estimate or predict the true breeding value of animals. And this is particularly the case where genomics has the major role when the accuracy of the EBV, which we can calculate from pedigree and performance information, is quite low. So such as when animals are young, so more for young animals where there's limited information that has been recorded on them. For traits which are difficult to measure, for traits that can't be measured prior to breeding age, for traits with a low heritability, and I guess in situations where it's difficult to collect effective performance information. So in smaller herds, situations where animals have been removed from their contemporary group prior to measurement. So particularly when, say, people are using yearling bulls within their program and they can't measure the full range of traits before they split that bull out and join him. So all the genomic information, the key role for us in using this information is to increase the accuracy of our breeding values. And importantly, I want to make this point as we talk throughout this presentation about the value of genomic information, that genomics doesn't replace the need to collect pedigree and performance information. The role of genomics in our genetic evaluations today is to enhance the accuracy of the EBVs that we get from pedigree and performance information. So it's very much an addition rather than a replacement of what we need to do to get indications or descriptions of the genetic differences between animals. Now, as genomics is a new technology and is continually evolving and has evolved a long way from when the first genomic information was included in the Angus BREEDPLAN evaluation back in 2011, so too is the software for incorporating genomic information into the calculation of EBVs. And if we look at the different methodology which is used to incorporate genomic information into Angus BREEDPLAN, there are several different pieces of technology or methodology which is used. Particularly in the case of the carving ease traits, we use a multi-step or a blending approach, which was the traditional approach used across all traits. But we now, across most of the traits where we incorporate genomic information, use a new methodology known as single-step technology. For some of the traits, we still, and some of them which are a little bit more difficult to analyse, so docility and the structural traits, no genomic information is currently included in the calculation of those EBVs. Now, this is obviously from an Angus BREEDPLAN evaluation. Similar type of situation occurs in the Hereford genetic evaluation, which is conducted by Herefords Australia and the New Zealand Hereford Association. As far as I'm aware, in New Zealand, they're the only breeds currently which are utilising genomics within their evaluations. So I want to focus now on the single-step approach, and the benefits of that. But firstly, if we go through and just review what the multi-step procedure was, which is the procedure, as I outlined, that is still used for carving ease EBVs, but also was traditionally used when it was first introduced in 2011. And we have a multi-step procedure for incorporating genomic information. So particularly the first step, if you like, in calculating the EBV with genomic information is to go back and calculate a traditional breeding value. So we have pedigree and performance data, which we put through the BREEDPLAN analytical software and calculate an EBV. The second step is we've got genomic data, which we utilise that. We put that through a genomic prediction equation, if you like, or a prediction equation, to come up with a genomic prediction or a genomic breeding value for the animal. And these were traditionally calculated by the genomics companies. So in the case of Zoetis, they were reported as our molecular value predictions, or MVPs. In the case of Neogen, or GeneSeq at the time, molecular breeding values, or MBVs. So we had two different sources of information. We had a traditional EBV based on pedigree and performance. We had the animal's genomic breeding value or genomic prediction. And then we used a process called blending, if you like, where those two bits of information were combined together to come up with a genomically enhanced EBV. So this is the procedure which has utilised since 2011 and has worked quite well for the Angus breed, but also the other breeds that were utilising similar technology. But as the software evolves for incorporating genomic information, there was a new approach known as the single-step approach, which was implemented into our genetic evaluation in late 2017. Now, the new approach effectively has one major change in that we no longer go through and calculate that traditional breeding value and the genomic prediction, but rather we put all the information into the mix, and in the one step, if you like, so we have our pedigree and performance data. We now have our raw genotype information, so not a genomic prediction, not an MVP as such, but the raw gene marker information, the raw SNPs, which we all put into the one pot, if you like, and all in the one step it goes through and calculates that genomically enhanced EBV. And the benefits, I suppose, there are several benefits of doing this approach and why it's been developed. The main benefit is how we model and estimate genetic relationships between animals within our genetic evaluation. So traditionally this has all been done through pedigree information, but now we have the benefit of being able to utilise some genomic information in estimating relationships between animals. Now, to explain that and the change in approach, we need to go back and review the manner, I guess, in which genes are passed from one generation to another. So if you like, on the screen here we have a sire and a dam. Hopefully most people are familiar with the fact that each parent possesses two different copies of the gene, and when one of those copies is passed to the offspring. So in the case of the female here, the two different copies of the gene, the particular gene, one is blue and one is green, and half the time the progeny receive the blue copy of that gene and half the time they receive the green copy of the gene. So as a result, we end up with progeny which basically receive a different, I guess, mix of genes from each parent. Now, based on what we know about that in terms of how genes are transferred from one generation to another, we can go through and actually estimate what the genetic relationship is between animals. So as we'll see here on the screen again, we can estimate that the genetic relationship, or another way of looking at it, is the genes that animals have in common between an offspring and its parent has a genetic relationship of what we would call 0.5 or 50%. Full siblings, so siblings from the same embryo transfer flush where they've got the same sire and dam, or just general full siblings, not necessarily through an embryo flush, but where they have the same parents, would have a genetic relationship of 0.5, and half siblings at 0.25, so where they have the same one parent in common. Now these relationships, I guess, was what has traditionally been used in the calculation of the EBVs. So we've used the pedigree information combined with this knowledge of how genes are passed from one generation to another to estimate the relationship between animals when calculating EBVs. And for those that are more technically minded, this is often referred to as the A matrix, which is where it fits within the software. Now in practice, however, we know that transfer of genes from one generation to another is a little bit more complicated than that. So there are a few other things going on that influence that. In particular, before the example we used, we were just looking at one gene. In practice, genes are not transferred in isolation, but as part of a chromosome. And likewise, when we exchange, and I guess inheritance takes in that different chromosome segments are kind of crossed over, if you like, or exchange of chromosome segments occurs during meiosis, during a process called crossing over, which means that when we actually look at it, while we had those standard relationships we were talking about before, there is some individual variation without them. So this example on the screen just shows the relationship, if you like, between full siblings. So before on the previous screen we showed that on average full siblings will have a genetic relationship of 0.5, whereas in actual fact there'll be some variation, and they may actually, the relationship between two full siblings may anywhere be, for example, 0.4 through to 0.6. So some siblings will be more closely related to one another than other full siblings. Now the real benefit of the new single-step methodology is we can actually start to now use the DNA information to better estimate the genetic relationship between animals. So in particular we can use our traditional pedigree A matrix approach where we use the pedigree information, but now incorporate some genomic information into that to more closely define the genetic relationship which is used in the calculation of the EBVs. And in particular, if you like, if we're not technically minded again, we have our A matrix. We now calculate a G matrix, if you like, of relationships, a matrix of relationships between animals based on their genomic information. Combine the A matrix and the G matrix into what they call an H matrix, which is the actual relationship which is used in the calculation of the EBVs. And the net effect of that is that rather than using just the same relationship between all animals, then we can now model for animals which have genomic information, we can start to model that relationship between individuals more precisely. And in particular, animals will now have different relationships based on their DNA information. So if we look at the relationship between a progeny and their parents, before it was a 0.5 relationship, now it will be more defined based on what the genomic information is telling us about their relationship. So what the implication for EBVs is that has is a couple of different things that we see. First of all, is there might now be considerably more variation in the EBVs of full siblings and half siblings. So that's calves from the same ET flush. Before they used to be fairly closely aligned. Now we can see far more variation within those. Likewise, progeny of the same sire used to be reasonably closely aligned. Now we can see far more variation within that progeny group. Likewise, in a similar vein, the EBVs of calves may be less aligned with the EBVs of their parents. Importantly, I suppose, the influence of any pedigree error that we may have is greatly reduced because we're now utilising the genomic information, and it will largely overcome some of the paper pedigree errors which might be in the system. Likewise, one of the other big key things we see now is we've got animals which might be completely unrelated by pedigree, but the genomics information would suggest that they have a genomic relationship. And so we start to see information on animals which are unrelated by pedigree start to inform the EBVs of other animals which they have a genomic relationship to. So this can have quite a significant effect on the EBVs that are calculated. Some other benefits of the new single-step approach, and as I said, the main benefit is how we model differences in the genetic relationship between animals. But genomic information now contributes to the EBVs for relatives as well as the animal for which the genomic information has been collected. So before, in the old blending approach, genomic information only contributed to that animal's own EBVs. Likewise, genomic information now calculates to all EBVs within the main multi-trait analysis. So before there were specific EBVs for which the genomic information was incorporated. Now it incorporates across all EBVs which are analysed in the main multi-trait component of the Angus BREEDPLAN genetic evaluation. One of the significant benefits, it won't be all that noticeable at a breeder level, but is the need to regularly recalibrate the prediction equations. So the equations which took the genomic information and calculated the genomic breeding value, before they had to be regularly recalibrated to make sure they remained relevant. And that meant that animals' genomic values had to be periodically updated from time to time. Now that all happens within the fortnightly BREEDPLAN analysis and removes that need, which means that the genomic information that's contributing to the EBVs is constantly updated and constantly giving the best possible information. Likewise, genomic, there's some better quality assurance, I suppose, around the incorporation of genomic information into the analysis. In particular, now genomic information will only be incorporated into the analysis and the calculation of EBVs if animals have sufficient relationship to the reference population, or the reference population being the population of animals within our evaluation that have both performance information and genomic information, which guide the contribution that the genomic information has to the calculation of the EBVs. And likewise, there's far more routine checks in there, checking things around the parentage of animals, so checking how accurate the pedigree is that has been recorded, using the DNA information to check the sex of animals which has been recorded, and also the breed of animals. So there's a lot more quality assurance which results from this single-step BREEDPLAN analysis. Now, the ultimate benefit of the new methodology is that we end up with EBVs which more accurately predict the breeding value of animals. And ultimately, when we're more accurately describing genetic differences between animals, more accurately identifying and utilising those animals which are most aligned with our breeding objectives, and resulting in greater genetic improvement being achieved within our breeding programs. So that's the new methodology as such, and hopefully it gives you a good overview of the change. I want to just finish now by giving some messages for seed stock producers. So what does the availability of this new technology actually mean to what people could be doing on farm? The first thing I need to preface this with is this is really applicable to Angus producers, also Hereford producers who have similar bits of availability of this technology available to them. For other breeds we may not yet be incorporating genomic information in their genetic evaluation, obviously this may not be as applicable to you. The first thing I want to make a note, I suppose, is to where does genomics fit and what does it mean for seed stock producers, is that genomics is part of the future. So genomics is now starting to become a more mature technology, and it really is a case now of all seed stock producers of beef cattle really need to be considering how genomics can be best applied within their enterprise. And that's no different to any of the other technology that's available, but the technology is here and it's here to stay, and it is offering quite a few benefits. So looking at how you might apply that within your breeding program is something that all seed stock producers should be doing. Now, by all means, that doesn't mean that everyone should be going out and genomically testing all their animals, but they should be starting to think about how they can get the best out of this technology. So some general recommendations which I would make, and they are very much general recommendations, so they may not apply to all individual circumstances. The number one point I'd make when you start looking at genomic information is really to stress that point that even though the technology is offering a lot of benefits, it doesn't replace the need to record pedigree and performance information. So performance information still remains king. If we're trying to get the most accuracy in the calculation of our EBVS, then recording as much performance information on animals for as many traits as possible is still the key ingredient to describing their breeding value. Having said that though, genomics now offers some advantages in terms of how we can improve or increase the accuracy of that information, and effectively get, one way of looking at it is that we're getting information on a few progeny before we would have otherwise. The second recommendation I would make is really, irrespective of whether people are actually genomically testing their animals, they should be looking to collect and store DNA samples on all their animals as part of a routine management practice. So whether that be at birth or at weaning, early on in their lives, early in the animals' lives, they should be collecting DNA samples. The easy thing, certainly from an Angus producer in Australia's perspective, is to collect a hair sample, take that, put that in an envelope, put the animal's identifier on the front, seal the envelope up and leave that stored on farm, and then you have the DNA resource available at any stage if you want to undertake any genotyping. The third recommendation I'd make is really, in my view now, you should be going through and genotyping all key sires and donor dams within your operation. So just as a normal process, when bulls are joined, prior to joining, if they haven't been genotyped, collecting a sample, sending that in and getting that genotype information. Fourthly, outside of those two recommendations to collect and store DNA samples and genotype key animals, I think it's really a case where all individual seed stock producers should be going through and developing a genomic testing strategy for their herd. Now that's going to vary from operation to operation, but really a genomic testing strategy regarding what samples am I going to collect, be it the different types of samples, what works best for you, when am I going to collect them, what genotyping, what animals are going to genotype, what products am I going to use, what are the considerations that I have with my breed society in terms of their requirements. Going through and carefully considering all those things to come up with a genomic testing strategy for your herd. And this technology is moving so quickly that it's really paramount to keep on top of what's happening and to regularly review and update that genomics testing strategy for your herd, and to ensure that you're getting the most out of the opportunities which are available to you. The last recommendation I would make, and this is probably from experience and seeing some of the pitfalls that people fall into, is really to go through, be organised, test well in advance, so undertake your genomic testing well in advance of when you need the results, because genomic testing does take some time, it does take a while for the results to come back. So being organised, getting everything organised well in advance will ensure that you can get the most out of genomics. So with that, I'll conclude for the evening. Hopefully that gives you a good insight into some of the benefits that are available from genomic testing, some of the developments which are available in the methodology in terms of how the genomic information is used in the calculation of EBVs. And certainly if anybody has any questions, then please don't hesitate to contact me via the phone number or the email address on that screen. Thank you.